Now we go for the next question. The following are the structures seen in the ommatidium of cockroach. Arrange them in the correct sequential order from anterior to posterior. Now here what are the options? Cone cells, retinular cells, Carnegian cells and optic nerve fibers. Now as we observe the body of cockroach, this part we call it as the head region and this is the dorsal side. Dorsolaterally one pair of what are present? The compound eyes. In each compound eye 2000 ommatidia are present. 2000 ommatidia. This is the unit of compound eye. Total number of ommatidia in both the compound eyes 4000 ommatidia. Now as we observe the structure of ommatidium, the ommatidium is elongated and pyramid-like. In the ommatidium, the first part we call it as the cornea. It is a biconvex lens. It is followed by Carnegian cells or the lenticular cells. And it is followed by the cone cells or vitrillae. And it is followed by the crystalline cone. Up to this you have to observe the arrangement. Cornea, Carnegian cells, cone cells and crystalline cone. This is one arrangement of question from anterior to posterior. Now the cone cells are followed by these are called retinulae. Number of retinulae 7. 1, 2, 3, 4. 1, 2, 3, 4, 5, 6 and 7. Now these retinulae secrete the rhabdome or means rhabdome. Now this is the ommatidium. Entire ommatidium based on basement membrane. The retinulae are nothing but the nerve cells. From these nerve cells, the nerve fibrils will arise. They unite to form one optic nerve. Now this is one compound eye. This is another compound eye. In each compound eye, how many ommatidia? 2000 ommatidia. From each compound eye, one optic nerve arises. Now this is optic nerve. Around optic nerve, the ommatidia are arranged radially. Now based on this information, you observe the answer. Arrange the parts of ommatidium from anterior to posterior. One is the cone cells and retinular cells, Carnegian cells, optic nerve fiber. In the given question, depending upon the options, how to arrange the structures from anterior to posterior. The first is Carnegian cells. The Carnegian cells followed by the cone cells. The cone cells are followed by retinulae and followed by the optic nerve fiber. Now we go for the next question. Type of image formed in nocturnal insects. Type of image formed in the nocturnal insects. Now for the nocturnal insects, one best example is cockroach. In the cockroach, what type of image is present? The superposition image. What type of vision? The superposition vision. Now here the V vision, here the I image. In nocturnal insects, superposition image as well as the vision is superposition. As we observe the ommatidium structure, this part we call it as cornea. It is followed by Carnegian cells. It is followed by the cone cells. It is followed by the crystalline cone. Now, this part we call it as dioptric region. This part we call it as dioptric region. The dioptric regions are separated by primary pigment cells. Now, it is followed by the 7 retinulae. 7 retinulae. And this one we call it as the rhabdome. In case of nocturnal insects, here in the Ommatidium, what are absent? The secondary. This indicates secondary. The secondary pigment cells are absent. In the brackets, this indicates absent. In the nocturnal insects, in the Ommatidium, secondary pigment cells are absent, as well as there is a gap between cone cells and retinulae. There is a gap between cone cells and retinulae, so that in the nocturnal insects, the Ommatidium not only receives the light rays coming through its cornea, but also absorbs the light rays from the adjacent Ommatidia. Again it is repeating. In nocturnal insects, there is a gap between cone cells and retinulae and no secondary pigment cells. Because of these two reasons, in nocturnal insects, each Ommatidium not only receives the light rays coming through its own cornea, but also absorbs the light rays from the adjacent Ommatidia. So, in each Ommatidium, though image forms, all images are overlapped. So, the image is not clear. The vision is not clear. It is blurred. Such type of vision we call it as superposition. Such type of image we call it as superposition. Now, type of image formed in nocturnal insects: mosaic, apposition, superposition, clear. Here the answer is superposition. Now we go for the next question. Assertion: respiratory pigment is absent in cockroach. Reason: tracheal system is develops which delivers the air directly to the tissues. Now, as we observe the respiratory system of cockroach, in cockroach, as we observe the RBC totally absent, and there is no hemoglobin, only WBC present. So, in the body of cockroach, the blood never delivers the oxygen. In order to get the oxygen, what type of system is present? The tracheal mode of respiratory system. Now, this is the spiracle, and this is the atrium, and this is the trachea, and this is the tracheal cell, and these are the tracheoles. Now, the tracheoles end in the tissues. What is meant by tissue? The group of cells. Now, this is the tissue. Because of the tracheal mode of respiratory system, in the body, each and every cell directly gets the oxygen. Now, the oxygen enters into the body, spiracle, atrium, trachea, tracheoles, and enters the cells of tissues. Now, assertion: respiratory pigment in the body of cockroach is absent. It is 100% correct, because no RBC, no hemoglobin. Reason: tracheal system helps to deliver the air directly to the tissues. So that both are correct or explains A. Now we go for the next question. These structures of Ommatidia jump into the action after each molting. Now again repeating the question. These structures of Ommatidia jump into the action of after each molting. Again repeating the question. These structures of Ommatidia jump into the action after each molting. Now as we observe the ommatidium, this part we call it as the cornea. The cornea is made by cuticle. At the time of molting, the cornea is removed. It is called molting. The cornea is followed by the cornigian cells. The cornigian cells are also called the lenticular cells. Now, the ommatidium externally covered by cornea. It is made by cuticle. At the time of molting, the cornea is removed. Whenever it is removed, the cornigian cells jump. It means the cornigian cells secrete the cornea. So that here the answer. These structures of ommatidia jump into the action after each molting. One is lenticular cells, cone cells, retinulae and rhabdome. Now, here what is the other name of cornigian cells? The lenticular cells. So, the answer is the lenticular cells. Now, we go for the next question. Assertion. The tracheal system of cockroach is called holoneustic tracheal system. Now, in the body of cockroach, how many spiracles are present? 10 pairs of spiracles are present. All these 10 pairs of spiracles are functional. If all spiracles are functional, it is called the holoneustic. Now, what is meant by polyneustic? What is meant by polyneustic? Out of 10 pairs of spiracles, if at least 8 pairs are functional, that condition we call it as a polyneustic. But in the body of cockroach, all 10 pairs are functional, so called holoneustic. Now, regarding this question, assertion. In the tracheal system of cockroach, the condition is holoneustic. All 10 pairs are functional. Now, reason. All these spiracles are kept open during inspiration and expiration. Now, as we observe the inspiration and expiration, in the inspiration, only thoracic spiracles open, the abdominal spiracles close. During the expiration, the thoracic spiracles close, whereas abdominal spiracles open. So that regarding the reason, all spiracles are kept open during inspiration and expiration. That is wrong. But the first one is correct. All 10 pairs of spiracles are working or functional, so called holoneustic. Now we go for the next question. The level of tracheolar fluid in the cockroach during cursorial locomotion. Now the question is the level of tracheolar fluid in cockroach during cursorial locomotion. Now this part we call it as spiracle. It opens into atrium. This is trachea. This is tracheal cell, and these are the tracheoles. Now this one we call it as the tissue. The tissue consisting the group of cells. In the tissues, what is present? The tissue fluid is present. Now as we observe the process of respiration, whenever the cockroach is active, what are the examples for the active? One we call it as the running. The cockroach runs very fast, it is called cursorial. The second one we call it as flying. Now, whenever the cockroach is active, the fluid which is present in the tracheoles re-enter into tissues. Whenever the cockroach is inactive, from the tissues, the tissue fluid enters into tracheoles. So, whenever the cockroach is inactive or at rest, From the tissues, the tissue fluid enters into tracheoles. But here the question is, the cockroach is active. One example is running. One example is flying. Whenever it is active, the metabolic rate is more and the demand for oxygen is more. So that from the tracheoles, the fluid again enters into tissue. So at the time of active, in the tracheoles, the fluid is very less. In the tissues, the fluid is more. Now, as we observe the question, the level of tracheolar fluid in cockroach during cursorial locomotion. One is more in tracheoles, second is more in tissues. Now you have to observe the diagram. Whenever the cockroach is active, from the tracheoles, the entire fluid enters into tissues. So in the tissues, the tissue fluid is more. So the answer is second. Now we go for the next question. The abdominal segments in cockroach which do not possess both nerve cord and spiracles. Now again, as we observe the nervous system of cockroach, this part we call it as the abdomen. In the abdomen, 10 abdominal segments are present. 10 abdominal segments are present. Now in the body of cockroach, in the abdomen, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 abdominal segments are present. Now in the abdominal region, the nerve cord extending up to 7th abdominal segment. Now this is first abdominal ganglia, second abdominal ganglia, third abdominal ganglia, fourth abdominal ganglia, fifth abdominal ganglia and sixth abdominal ganglia. Now what are the abdominal segments in which nerve cord present but ganglia absent? Now it is represented as a star mark. Now this is fifth abdominal segment. In the fifth abdominal segment, nerve cord present but no ganglia. Now in the 8th abdominal segment, 9th abdominal segment, 10th abdominal segment, both nerve cords and ganglia absent. Now here the abdominal segments in the cockroach which do not possess both nerve cord and spiracles, the answer is 8th, 9th, 10th abdominal segments. Now the next question. In cockroach, the respiratory pigment is, one is hemoglobin, second one hemocyanin, third one chlorochlorine, fourth one absent. Now as we observe the body of cockroach, in the body of cockroach no RBC, no hemoglobin. So in the body of cockroach, the respiratory pigment totally absent. What about the hemoglobin? It is present in the annelids in the dissolved state. Hemocyanin, it is present in the class Crustacea and Arachnida. It is a copper containing respiratory pigment. Chlorochlorine, that is also seen in annelids. Now in the body of cockroach, no respiratory pigment absent. Now go for the next question. In cockroach, the opening and closing of spiracles is influenced by. For this question, we have to explain two answers. Now in the given options, carbon dioxide tension in the hemolymph, oxygen tension in the trachea, oxygen tension in the hemolymph, carbon dioxide tension in the trachea, carbon dioxide tension in the body wall, oxygen tension in the hemolymph, concentrations of carbon dioxide HO2 in the body. Now the question is, in the body of cockroach, opening and closing of spiracles. Now this one we call it as spiracle and this part we call it as valve. All spiracles are valvular. The opening and closing of spiracles is based on, one is the concentration of carbon dioxide and oxygen. This answer also correct. And the second one we call it as carbon dioxide tension in the hemolymph and oxygen tension in the trachea. So both the answers are correct. Now for this question, in the given options, one option is correct. But in reality, for this question, the closing and opening of spiracles based on concentration of carbon dioxide and oxygen, as well as carbon dioxide tension in the hemolymph and oxygen tension in the trachea. Now, go for the next question. The food canal in female mosquito is formed by. Now, as we observe the mouth parts of insects. Now, here the question is, the food canal in the female mosquito is formed by. Now, this part we call it as the head of mosquito. Dorsolaterally, what are present? The compound eyes. These are called antennae. Now, this one we call it as the labium or proboscis. Now, this one we call it as the labrum. It is elongated. And this one we call it as epipharynx. This one we call it as epipharynx. Now, this one we call it as the hypopharynx. In the hypopharynx, what is present? Salivary gland duct. Now, in the female mosquito, the food canal or food tube is formed by. The answer is the labrum, epipharynx plus the hypopharynx plus salivary gland duct. Now, as we observe the options, the food canal in female mosquito is formed by labrum epipharynx labium, hypopharynx proboscis, labrum epipharynx hypopharynx. The hypopharynx included salivary gland duct. So, the third answer is correct. Now, we go for the next question. The mouth parts of CC fly. As we observe the sanguivorous insects. What are the sanguivorous insects? The blood sucking insects. One example is the female mosquito. One example is male and female bedbug. One example is the male and female head louse. One example is CC fly, etc. All these are blood sucking insects or sanguivorous insects. In the adult stage, in these insects, what type of mouth parts are present? Piercing and sucking. Whereas in the larval stage, what type of mouth parts are present? Biting and chewing. But here one exception is present. The lac insect, it is purely herbivorous. Though it is herbivorous, in the body of lac insect, in the embryonic stage as well as in the adult stage, what type of mouth parts are present? Piercing and sucking. Here the question is the mouth parts of CC fly. That means in the adult stage, biting and chewing, siphoning, sponging and sucking, piercing and sucking. The answer is piercing and sucking. The next question. The triangular plate-like structure in the mouth parts of moth. Now, as we observe the adult stage of moth, it may be silkworm moth or butterfly. Now, as we observe the siphoning type of mouth parts, this part we call it as head region. Dorsolaterally, what are present? The compound eyes, and these are called antennae. And this is the labrum. Labrum. It is very much reduced. Here the question, what is the triangular plate-like structure in the mouth parts of moth? In the moth, siphoning type of mouth parts are present. In the siphoning type of mouth parts, what is the triangular plate-like structure? That we call it as the labium. The labium having three segmented the labial pulse. Now, in the body of moth, what type of mouth parts? Siphoning type of mouth parts. In the siphoning type of mouth parts, what is the triangular plate-like structure? Labium. Attached to the labium, three segmented, one pair of labial pulses are present. Now, the answer is labium. We go for the next question. the proximal part of labium in the proboscis of housefly. Now, as we observe the housefly, in the housefly, sponging and sucking type of mouth parts are present. Now, this part we call it as the head region. This is head region. And these are called the compound eyes. And these are called the antennae. In the head region, what is the last scleroid? Clypeus. Attached to the clypeus, what is present? The proboscis. This entire part is called proboscis. The proboscis is modified labium. In the proboscis, the first part we call it as rostrum. Second part we call it as ostellum. Third part we call it as labellum. Now in the proboscis, what is the proximal part? Rostrum. What is the middle part? Ostellum. What is the distal part? Labium. Here, what is our question? As we observe the labium, what is the proximal part of labium? Again repeating the question. What is the proximal part of labium in the proboscis of housefly? This is proboscis. In the proboscis, middle part is ostellum. The ostellum is also said to be proximal part of labium. So here the answer is ostellum. Now we move for the next question. Assertion. The biting and chewing type of mouth parts are considered to be the most primitive. Yes, here the assertion is correct, because in almost all insects, as we observe their larval stage, in majority of insects, in their larval stage, the biting and chewing type of mouth parts are present. So they are the most primitive. In the biting and chewing type of mouth parts, what are the prominent parts? The mandibles. Hence they are called mandibulate type. Now the reason. The larvae of majority of insects have biting and chewing type. So A and R are correct. R explains A. Here one exception is present. As we observe the beneficial insects, in the beneficial insect, one example is a lac insect. No doubt it is a purely herbivore. In the lac insect, at all stages, in the larval stage as well as in the adult stage, what type of mouth parts are present? Piercing and sucking. That is one exception. Now we go for the next question. Again assertion. Insect mouth parts are provide a good example for the homologous organs. Now, what are the homologous organs? As we observe the homologous organs, now as we observe the homologous organs, the study of homologous organs, we call it as homology. All homologous organs derived from a common ancestor. All homologous organs having the similar origin. Homologous organs structurally similar but functionally dissimilar. The homologous organs explain the adaptive radiation. The homologous organs explain the divergent evolution. Now again repeating. What are the homologous organs? Structurally similar, functionally dissimilar. Homologous organs having common ancestor and the similar origin. Their study is called homology. They explain adaptive radiation. They explain divergent evolution. Here the assertion. The insect mouth parts provide a good example for the homologous organs. It is correct. The reason, the basic structure of all insect mouth parts is one and the same. As we observe the piercing and sucking, sponging and sucking, siphoning and lapping and chewing, all these mouth parts are fundamentally or basically derived from the biting and chewing type of mouth parts. So that both are correct or explains A. Now we go for the next question. Now what is the next question? Which of the following have biting and chewing type of mouth parts? Again repeating, which of the following have biting and chewing type of mouth parts? Now here one option we call it as the CMEX, and the second one we call it as the larva of silkworm moth. Now the third one is the CC fly, and the fourth one is larva of the butterfly, and the fifth one is the larva of the housefly. Now these are the given options where we are going to observe the biting and chewing. Now as we observe the CMEX, this is adult insect. In the adult stage, in the CMEX, what are present? Piercing and sucking. Now larva of silkworm moth. What is the larva of silkworm moth? That we call it as the caterpillar. In the larval stage of silkworm moth, biting and chewing. CC fly, this is adult stage. In the adult stage of CC fly, what are present? Piercing and sucking. Now the larva butterfly. The larva butterfly is called caterpillar. It has biting and chewing. Now the larva housefly, it is called maggot. What type of mouth parts? Biting and chewing. So regarding this question, where we are going to observe the presence of biting and chewing in the larval stage of silkworm moth, larval stage of butterfly, larval stage of housefly. Now we go for the next question. The number of joints in the maxillary pulps of housefly. Now as we observe the mouth parts of housefly, this part we call it as the head region, and this is called clypeus, and these are the compound eyes, and these are antennae, and this one we call it as the rostrum, and this one we call it as ostellum, and this one we call it as a labellum. Here what is the question? The number of joints in the maxillary pulse of housefly attached to rostrum, what are present? The maxillary pulse. Now as we observe the maxillary pulse, in the maxillary pulse no joints. Now based on this information, the number of joints in the maxillary pulse of housefly is: one is unjointed, second 2, third 3, fourth many. But here in the maxillary pulse no joints. So the answer is the first one. Now we move for the next question. The lifespan of workers, where we identify this question. As we observe the beneficial insects, one we call it as honeybee. As we observe the honeybee, 3 categories are present. One we call it as queen. Its lifespan is 5 years. As we observe the drone, it is also called male honeybee. The lifespan is short. After completing the copulation, because of the starvation, the male honeybee dies. Why the male honeybee dies? In the male honeybee, the mouth parts are reduced, so that after copulation, because of the starvation, male honeybee dies. What is the leftover? That we call it as worker honeybee. Now, what are the options? 5 years, that is concerned with queen honeybee. Second, 2 to 4 months, that is concerned with worker honeybee lifespan. What about the very short period? Concerned with the male honeybee. So regarding this question, what is the correct answer? the 2 to 4 months regarding the worker honeybee. Now we go for the next question. Extracting silk thread from the cocoon. Now, as we observe the second beneficial insect, that one we call it as silkworm moth. In the life history of silkworm moth, this one we call it as the larva. The larva is called caterpillar. Now the larva coils the silk thread around its body. This is silk thread. Now the larva plus silk thread, we call it as cocoon. The cocoon is also called the seed. Now from the cocoon we are extracting the silk thread by a process called the reeling method. Now as we observe the options and question once again, extracting, that is removing the silk thread from cocoon, is one is stifling. What is the meaning of stifling? The process of killing a pupa is called stifling, that is eliminated. Third option, recycling, that is eliminated. Fourth option, ginning, that is eliminated. Now from which stage we are extracting the silk thread? That is cocoon. From the cocoon we are getting the silk thread. The process of removing silk thread or getting silk thread, that we call it as reeling. It is done by using reeling machines. Now we go for the next question. Study the following. This is a linked question or 3 columns matching. Now you observe the first column. What is the insect? Silkworm moth. Second, lac insect. Third, housefly. Fourth, headlouse. Second column, number of eggs. Now the silkworm moth, the female silkworm moth releases 300 to 400 eggs. It is correct. In the life history of silkworm moth, the larva undergoes how many moltings? 4. That is correct. Now second, lac insect. The female lac insect releases how many eggs? 200 to 500 or hundreds of eggs. It is also correct. In its life history, the larva undergoes how many moltings? 3. Now the third one, housefly. The housefly, female housefly releases how many eggs? 500 to 600. In how many batches? 5 or 6. For each batch, maybe 100 or 125. Now the larva of housefly undergoes how many moltings? 2. Now the last one is headlouse. As we observe the headlouse, the female releases how many eggs? 80 to 100. Now as we observe the female headlouse per day releases 3 to 7 eggs. How many days it releases eggs? 30 days. Now per day 3 eggs. 30 into 3, 90. Or per day 7 eggs. Now as we observe the female headlouse releases how many eggs? In 30 days, 80 to 100 eggs. Now in its life history, the larva undergoes how many moldings? 3 moldings. So all these 4 are correct. Now the correct answer is 4th one. Now we have to discuss some bits orally. Now as we observe the remaining part of cockroach, what are the repeated questions in the cockroach digestive system? As we observe the body of cockroach, in the body of cockroach, in the digestive system, the alimentary canal classified into one is foregut, second one midgut, and the third one we call it as hindgut. Now the foregut, hindgut, these two are derived from ectoderm, whereas the midgut is derived from endoderm. Now as we observe the cockroach, what is the larval form of cockroach? The nymph. Now what is the larval form of bedbug? The nymph. The larval form of headlouse, nymph. In the next class we have to discuss the remaining topics with important bits.